Do you look at your DNA match list on Ancestry or MyHeritage and wonder, what do these numbers actually mean? Take Orla here, who I match with on Ancestry. We've got this number of 95, C capital M, and if I click into Orla's page, it's giving me a little bit more information that it's 95 cm across 4 segments. What does that mean? Let's start with the basics, the measurements that we're working with. This number here, the cm or centimorgan, think of it as the unit of genetic relationships. That's how we measure how much DNA we have in common with somebody else. I share 51 centimorgans with Nicole. But here's the thing you have to get straight. It's not physical length, it's not an inch or a centimetre. It's a measure of probability. It's a unit that measures the chance that a chunk of DNA got passed down to you from an ancestor completely intact. Take this illustration where the sibling on the left has inherited half their DNA from their father, half their DNA from their mother. But if you take a single chromosome, chunks of DNA have got mixed up during the process of inheritance. We have this light section here, which represents one chunk of DNA, but then a different chunk of the father's DNA comes to this smaller section, and then this much tinier chunk of father's DNA from a different chromosome comes here, and so on and so forth. It's a jumble. Every generation, our DNA gets shuffled around like this in a process called recombination. So the bigger the centimorgan number, the bigger the chunk of DNA, and the less likely it was to get broken up by all that shuffling. And that usually means it came from someone more recent in your family tree. Here's a common analogy. The number of centimorgans isn't about distance to a destination, it's about the time it takes to drive there. Let's say your destination is a mile away. If you're travelling on an empty road, it's going to take about a minute. But suppose you've hit peak commute time in a city centre. Your journey could take 20 minutes. Same distance, but different terrain. Our DNA is like that. Some parts of our chromosomes are like those busy city streets. They are recombination hotspots where shuffling happens all the time. Other parts are like those empty stretches of open road. Centimorgan is like the drive time. It holds more insight into the relationship than just the distance. So who came up with this unit? The story starts over 100 years ago, and it has nothing to do with humans. Back in 1909, a 19-year-old undergraduate named Alfred Sturtevant joined a renowned biology lab at Columbia University. The researchers were investigating the effects of breeding multiple generations of fruit flies. Of course, these little critters live only a short time, so you could research fast. Alfred and the team noticed something strange. Some traits, like eye colour and wing shape, seemed to be linked. They were almost always inherited together, but every now and then they would swap places. Let's say from generation to generation that red eyes and straight wings normally go together, while white eyes and curly wings normally go together. Occasionally, the chromosomes cross over and swap segments, and this produces new combinations. So you get red eyes with curly wings and white eyes with straight wings. Alfred has an aha moment when he asks himself, what if how often the traits swap places tells you how far apart they are on the chromosome? Genes that are far apart have a lot of space between them to get swapped during that recombination shuffle, but genes that are close together tend to stick together. Alfred drew the world's first genetic map, and he needed a unit of measurement, which is now our centimorgan. You may be wondering why it's not the Alfred. His professor was Thomas Hunt Morgan. So now we know what a centimorgan is and where it came from, but the importance isn't about the total number or size. It's not just about how much DNA you share with someone, it's how you share it. Let me explain with another analogy. This one is a bit more poetic license, but bear with me. Let's say you're at a party with 2 tables of food. Table A has several large pieces of biscuit cake. Table B has lots of tiny crumbs that technically add up to the same amount. You're heading to table A, right? Because those big pieces of delicious biscuit cake are just better. It's the same in genetic genealogy. We look for the big pieces. They're just better. Okay, that analogy could use some work. So let me switch to the actual topic at hand. The one big, long, and broken segment of DNA is a massive clue that it came from a single, pretty recent common ancestor. It's solid evidence. All the tiny little crumbs, the little segments that add up to the total, they could be random noise, or they could be tiny hand me downs from dozens of different ancestors who lived hundreds of years ago. So here's the important point. One big segment is worth a lot more genealogically than a whole pile of little ones. Hey, that cake analogy is making a bit more sense now, right? Let's run through some common complications you may face with interpreting your DNA matches. First is when multiple relationships are involved. You can be related to someone in more than one way. Let's say your great grandparents were cousins. Then that shared DNA just gets added up, and that makes your relationship look closer than it really is. You may be third cousins, but the number of centimorgans that you share is more within the range of second cousins. Second complication, endogamy. It's the same deal here, really. If your ancestors came from a community that married within itself for generations, you share tons of little DNA crumbs with everyone. It's like the table with shattered biscuit cakes on it, loads of crumbs that inflate your total CM number. The third complication, genetic variation. Remember that genetics is all about chance. Even you and your full sibling didn't get the exact same DNA from your parents. So the numbers are always a range, not one perfect number. I'll finish on a fourth complication that you'll likely meet if you test your DNA with two different companies, or as many people do, download your DNA from Ancestry and upload it to another company like MyHeritage, Family Tree DNA, and there are another few companies. I popped over to Reddit for just one example of someone complaining about a situation that pops up from time to time. So this particular individual is saying, I've been discussing with a DNA match recently, and we discovered that while we appear as close matches on MyHeritage, sharing 103 centimorgans over 6 segments, on Ancestry, we apparently only share 43 centimorgans over 5 segments. Has anyone else seen this before? Both of us took our Ancestry DNA results and uploaded them to MyHeritage, so they should be identical, right? He confirmed seeing the same numbers from his end, so I'm confident the two profiles belong to him. Bit of a head scratcher, right? But two people should be the same DNA, two different companies. And it's not just a small difference between the numbers, there's a massive gap. How can the results be this different? Which one do you trust? Or do you just trust neither? Let's address the differences between testing companies. It all comes down to the fact that they have different philosophies and different computer programs with how they count up centimorgans. My analogy now is a sieve. Ancestry discards or throws out tiny little segments, anything under about 6 centimorgans, because they consider them to be just noise. So they let them drop through the sieve. My heritage has a different sieve. It has a sieve that retains the tiny little segments. That makes the my heritage number collected in the sieve bigger. The second reason is how both companies handle what's called pile-up regions. These are like genetic traffic jams that city centre commute. There are bits of our DNA that are very common across entire populations. You may share this segment with someone, but the common ancestor is hundreds of years back and it's been passed down intact to all the descendants. Ancestry has an algorithm that tries to clear out the noise of the pileup regions. They call it timber. MyHeritage has a philosophy of showing you everything. So, which company is right? You could argue all day on that, and people do, but it's just two different outlooks. Ancestry's number is more conservative, so it's less likely to lead you down a false rabbit hole. But the company results are more likely to eliminate potentially genuine DNA matches that MyHeritage may throw up. The main takeaway is that building out your family tree is about genealogy. Always confirm your DNA matches with good old fashioned genealogical research. Right, and there you have it. If you enjoyed this video, please leave a like, consider subscribing, leave a comment. Hope this helps with your family research.